So for today's practical, we're going to learn how to use pipettes. We're going to be using 3 pipettes for today's practical. These are the P20, shown here in yellow, the P200, again shown in yellow, and the P1000, shown here in blue. So these pipettes are used for measuring very small volumes. The P20 is used for measuring between 2 and 20 microliters. The P200 is used for measuring between 20 and 200 microliters. And the P1000 is used for measuring between 200 and 1000 microliters, or 1 mil. We'll be using these pipettes in order to measure out small volumes during the practical using 2 different methods. So for this part of the practical, we're going to be pipetting 500 microliters of water as an example. For this, we're going to need a P1000 because it falls within the range of 200 to 1000 microliters. So the first step is going to attach a pipette tip, which are blue, which matches the color of the actual pipette. The second step is to set the volume that's required on the actual pipette. And if you take a look at the actual dial, you can see that we're changing the volume to 500 microliters. So once the volume is actually set, we can then begin to transfer the liquid from one container to another. So the first step is to push the pipette barrel down to the first stop, insert it into the container of liquid, slowly depress upwards, which takes up the volume into the actual pipette tip. The pipette tip holds the actual volume of water. It is then transferred into the second container, which in this case is an Eppendorf, and then slowly the top button is pressed downwards to the first stop. After a second or two, the second stop then is reached by pressing downwards again, and the final volume of liquid is released into the actual Eppendorf. For the first part of the practical, we're going to need some materials. These are the pipettes, the P1000 pipette, which is the blue one we've introduced already, a P200, which is the yellow one we introduced already, their associated tips, blue for the P1000, yellow for the P200. We're going to need a container of liquid and a microtiter plate. A microtiter plate holds very small volumes. It is 96 wells. It has 8 rows and 12 vertical columns. In this experiment, you're going to be pipetting some small volumes of liquid into the columns and carrying out replicate pipette measurements. For the first part, we're going to be pipetting 100 microliters. For this, we've chosen a P200 because it falls within the range of 20 microliters to 200 microliters. The first part is to set the actual volume using the dial. Once that has been set to 100 microliters, you can now put on the pipette tip using a yellow pipette tip which matches the color of the actual pipette. The first part then is to pipette in the volume of liquid, so 100 microliters, again using the technique demonstrated before, and pipette it into the actual well. So very slowly pipette out the volume of the actual liquid into the well, first going to the first stop and then finally going to the second stop. You'll repeat this a number of times throughout the whole column of the actual microtiter plate. So there are 12 wells in an actual column. Each of these 12 wells will be filled with 100 microliters. For column 2, it's a repeat of column 1. For column 3, we are now asked to pipette in 200 microliters. So again, setting the volume to 200 microliters and then pipetting into each individual well the volume of 200 microliters. For column 4, you're asked to pipette 300 microliters, but by a particular technique. Normally for 300 microliters, you would be asked to use a P1000 as it falls within the range of 200 to 1000. But in this instance, you're asked to pipette 300 microliters by pipetting twice the volume of 150 microliters into the same well. So set your pipette to 150 microliters and use this twice on individual wells of the same column. Finally, once all the volumes of liquid have been added to the microtiter plate, it is then placed into a spectrophotometer. The spectrophotometer is an instrument that absorbs light at particular wavelengths. In this case, we're going to measure the absorbance of light at 450 nanometers. This has already been preset for you on the machine and on the computer program associated with it. The plate is placed with position A1 in the top left-hand corner of the machine. Okay, so on the microtiter plate you'll see that the rows are labelled with letters and the columns are labelled with numbers. So the position A1 is in the top left-hand corner, and this is placed in the top left of the actual spectrophotometer. Once the absorbance is calculated by the machine, you will get a printout on the computer for your results. You will then be asked to calculate the mean, standard deviation and coefficient of variation for each column and record the results in a table provided in your lab manual.